My name is Ashley Volz. I'm here on site at the RNA Leaders event in Basel, and I'm here speaking with Bernard Sagaert, CEO of etherna. Thanks for being with us today.
Thanks for inviting me, Ashley.
Let's get right to it. What are the biggest challenges to resolve for the next generation nucleic acid-based medicines?
Well, if you would ask the whole audience here, probably everybody will say delivery, delivery, delivery. And from our perspective, being in the space already that long, it's a lot more complicated than that, because we see that if you want to bring the RNA modalities to the next stage, you actually need to start building on different angles. If you want to go to protein replacement, for instance, you need to have higher doses. Higher doses means you need to have your safety a lot better under control. It's also intended for lifelong administration, so you need to be able to redose multiple times. So it means that your whole design of your product needs to be differentiated to what we currently have. What we currently have is, in commercial, is infectious disease, so prophylactic vaccines. So you want to have a immune response to your LNP, to your payload. Once you get to protein replacements, you absolutely do not want to have that, because if you get an immune response, you will actually reduce the effect of your medicine. So you need to get rid of that. If you want to go to autoimmune diseases, you want to have your formulation to have an opposite effect, actually reduce the immune response in the body. So those are the things that we will move into from the new therapeutics that will come. And so the whole modality around the payload, the RNA, the purity of the RNA, the ionizable lipid, the toxicity of the ionizable lipid, all of that needs to be redesigned, and that's what we're currently doing.
And what are your thoughts on the scalability of manufacturing processes?
Well, what has been done is everybody scaled up a lot the manufacturing of RNA and LNPs. What we have done was just the opposite. We improved the quality of the very smallest scale that we made, like 1 milligram of formulated RNA, and we made sure that we got to the right quality, so that we did not activate the immune system by the design of the RNA, by the design of the process to make the RNA. But we did that having in mind also the scalability. And so our processes are perfectly scalable, because they don't take into account certain special things that might not be scalable. No, the process itself is scalable, and that's how we look at it. And scalability is not about size. You first need to have your controls on the small scale and then scale it up. And that's what we have been focusing on.
And how does etherna differentiate from other companies in resolving challenges in your field?
Well, we have the RNA, we have the LNP, we have that integrated in one company together with a manufacturing branch. And I think nobody out there has the same thing, except for maybe a BioNTech, a Moderna that are keeping everything in-house. We are offering all of our know-how, all of our IP is offered in a service model way. Of course, with the licensing branch, that's we need to get some money somewhere, but it's offered in a collaborative way to partners, so that we can actually bring products to the clinic and commercial that are optimized in a certain indication. Because we see that every change that happens needs to be validated, and one of the changes that you naturally have in the field is that your payload changes. So you need to optimize your platform again with that change of a payload. And that's where we are differentiated, because we want to collaborate, we want to really bring products together with the customer, together with a partner. We don't call them customers, we call them partners. Want to bring them together with them to the clinic and then beyond.
Oh, sounds good. Well, thank you so much for being here and speaking with me today.
Thank you. Thanks for the interesting questions.